commercialization aspect of the theme one, which is basically marketing. I think as you heard this morning, you can grow all the Bitter-Gourd as you want, and you can really claim a lot of health benefits, but if you cannot market or if you cannot convince Jackie to eat bitter melon in 5 years down the road, I think we're not doing a good job. So I think it's an important theme to address this issue, and I think we have a good number of speakers here, and I'm going to follow some ground rules, make sure that the speakers finish their assigned time, otherwise we'll have 3 minutes and 1 minute and then there will be a stop. So please watch. If you're really sensitive on the time, try to watch at the end, and then we'll stop that. So the first speaker is Dr. Andreas Ebert. He's the Genebank manager at the AVRDC World Vegetable Center in Taiwan. And because his resume is here, I'm not going to read, so I will give that time for him to use that for his talk.
Good morning, ladies and gentlemen. I would like to open this presentation with a question to all of you. Is Momordica carantia of Asian origin? And all of you of this opinion, please raise your hand. Only 1, 2, 3, 4? Not more? Okay. I think this is really an interesting question, and I just would like to point out to this recent presentation, recent publication on the origin of Momordica, published in 2010 in Molecular Phylogenetics and Evolution, indicating that dated biogeographic analysis suggests that Momordica originated in tropical Africa and that the Asian species are the result of one long-distance dispersal event about 19 million years ago. So many things really point to the fact that Momordica originated in Africa. And well, while this paper indicates 47 species originating from Africa plus 12 in Asia and Australia, we could retrieve only 36 species based on GRIN USDA data. And if you look at this list of, here we have the cultivated species, Momordica carantia, plus all the other wild relatives, and here you see the map of Africa, and you see there's really a huge concentration of wild Momordica species in eastern, southern Africa, and then widely distributed over West Africa. And only a few wild species like Momordica cochinchiniensis, native in Asia, Australia, Southeast Asia, another Momordica dioica, native here in India, Pakistan, Sri Lanka, and Momordica subangulata, native in China, Bangladesh, and some Southeast Asian countries. Now, if you look at what is in our gene bank at AVRDC, we are capturing relatively good diversity of cultivated Momordica, but very little of wild relatives of Momordica. We have only 4 species in our collection, and you have seen there are more than 40 different wild species of Momordica existing. And I'm asking myself, who is really taking care of, who is conserving, and who is maintaining the genetic diversity of this crop originating from East and West Africa? Is it well captured in African gene banks? Would be a question, for example, to Tanzania or to West Africa, because these genetic resources, the wild relatives of Momordica, might play a major role in the future for breeding purposes. And as pointed out by Dr. Garwinkel, our collection, I think, going back to the origin of our center being an Asian vegetable research and development center, really was focusing on collecting germplasm from Asia. India has the highest representation in our collection, followed by Philippines, Thailand, Bangladesh, and Cambodia and Taiwan. Recently we did some collecting in Taiwan and focused on Momodica Carrantia Variedad abbreviata. And we have an interesting wild Bitter-Gourd collection from Taiwan in our gene bank. And just recently I found this publication indicating that this botanical variety is effective against inflammation. Now looking a bit at the diversity of cultivated Bitter-Gourd, we have different varieties in terms of fruit form, fruit shape, elongated, round, but I won't spend much time on this because Dr. Dillon, I'm sure, will go into much more detail on the cultivated Bitter-Gourd varieties he is using in his selection and breeding process. So now switching over to a bit of research we are doing on Bitter-Gourd storage, and the big question for us is Momodica seed really orthodox, or is it a recalcitrant species? We can observe that it can easily be dried to low moisture content, to 4 to 6% or even lower, without losing the germination ability. It can also be stored at relatively low temperatures of 5 to 10 degrees centigrade, but as soon as we store the seed under freezing temperatures, then the seed is basically dying. So we started a small collaboration with the University of Hohenheim in Germany, looking into overcoming conservation and germination problems in some selected traditional vegetables. Among those is Bitter-Gourd, and we looked at the impact of seed drying methods, seed moisture content and storage temperature on the germination rate of Bitter-Gourd. We did this together with the Institute of Plant Breeding and Seed Technology Research of this university. We selected 2 accessions, quite different accessions from our gene bank. One which originated from Thailand with small fruits and relatively low 100 seed weight, as compared with another variety from India, which is more of a commercial type. I would consider this a bit semi-domesticated variety compared to this variety from India. And if you look at the seed, you see the clear difference in seed size, seed form between these 2 varieties. So we have been growing, multiplying seed in net cages in the field. The variety from Thailand took much longer, has a very long growing period compared to the variety from India, but at the end we were able to harvest sufficient fruit and extract sufficient seed for our experiments. So right after harvest we extracted the seed, then washed the seed under running water, removed the surface water with the help of a centrifuge, and then dried the seed under shade for several hours, and then applied 3 drying methods using silica gel, drying beads, and the standard process used in our gene bank, dehumidified seed drying at 18 degrees centigrade and about 10% relative humidity. And the seed were dried to 3 target seed moisture levels of 4, 6, and 8%, and then packed into aluminum foil bags. And part of the seed was shipped to Germany, where each treatment combination was stored at 5, 10, 15, and minus 20 degrees centigrade for 6 months. At AVRDC we used seed dried to 6% SMC and stored the seed for 6 months. This is our drying room where we dried the seed, and our seed packing room. And then we used this Hycolab moisture meter to measure the equilibrium moisture content of seed with the surrounding air. It's a non-destructive method, and I think much better than the other destructive methods because we can reutilize the seed. Some preliminary trials which were done at the University of Hohenheim looked into the determination of seed germination of these two varieties, and the seed were submerged in water for 24 hours, and then the germination process started, and the seed were incubated at alternating temperatures of 25 to 30 degrees with 16 hours dark and 8 hours light. The first counting started at 3 days. Second trial was done without pre-treatment, but the result was not satisfactory in terms of germination. So the group looked into if the seed is really viable, and therefore usually the tetrasodium chloride test is used at 1%, and the standard treatment gave only a viability rate of 43 and 67% for the two varieties, so was really quite low. So another trial followed using cutting the seed into half with vertical and lateral cuts and removal of the seed coat, but still the coloring of the seed was insufficient. So in a third trial, similar methods were applied, and then vacuum was applied to infiltrate the solution to the seed, but still the result was unsatisfactory. Then freezing was applied and incubation, and still unsatisfactory. And then finally, with removal of the seed coat and the top 50% of cotyledons removed, and the remaining cotyledons cut superficially up to the proximity of the radicle, and following with incubation, vacuum infiltration and incubation for 4 days, then we really observed 95% viability of the seed. And this can be shown here. So with initial methods, normal standard practices, there is no full coloring of the seeds, but once the seed is cut horizontally, vertically, and at the sides, then we have full coloring, and the seed germination takes place. 95% of the seed germinated with seed coat attached or without, so this didn't make any difference. Now briefly mentioning our trials at AVRDC, where we used two storage temperatures, 5 degrees and 15 degrees centigrade. We didn't want to go into sub-zero temperatures because we knew that the seed would be dying. We stored the seed for 6 months and did some priming treatments before going into the germination process. Control, no treatment, 24 hours soaking in water, partial removal of the seed coat followed by soaking in water, soaking in household vinegar for 2 hours, soaking in potassium nitrate solution for 1 hour, and then again soaking for 24 hours in water followed by drying and drying for 72 hours in the humidified drying. We use this between paper method as recommended by ISTA, used alternating temperatures of 20 to 30 degrees centigrade and 8 hours light, and did a counting at 7 and 14 days. We looked into normal seedling development and abnormal seedling development and dead and hard seed percentage. We also did germination test in the greenhouse to see if there's any difference between incubator germination and germination in the greenhouse. And for the greenhouse, we used also 4 replicates with 25 seeds per treatment and did a counting at 20 days after sowing. And if you look at this table, Here we did a germination test of the fresh seed before drying, and you see a huge difference between the two varieties. Only 9% of the seed germinated of this variety from Thailand, while 73% germinated of the variety from India. And after drying, germination process slightly increased to 16% for this Thailand variety, and soaking in vinegar, for example, significantly increased viability, germination rate of this seed, and then also soaking in water plus 70 hours drying. For the variety from India, we had similar germination rates between fresh and dried seed, and most of the priming treatments increased the germination rate. Now, looking at the storage at 15 degrees and 5 degrees centigrade, we see this variety from Thailand had still quite a low germination rate of 25 and 42%, and germination was increased by scarification. We had a much higher and not differing germination rate between the two storage temperatures, and some of the priming treatments improved germination, and there were slight differences when we looked into germination under greenhouse conditions. But for this variety from India, basically the germination rate was 100%, so we were not able to increase germination through priming treatment. So in conclusion, we can say that the storage behavior of Bitter-Gourd seed is neither completely orthodox nor recalcitrant. Genotypes clearly differed in their rates of germination independent of storage and priming treatment, and seed storage at 5 degrees did not reduce seed viability when tested in an incubator. However, the accession from Thailand showed reduced germination rate under greenhouse conditions. In general, 6 months of storage at 5 degrees improved the germination rate, indicating that seed dormancy can be partially overcome by low temperature storage. And several priming treatments which can easily be applied by farmers enhanced seed germination. With this, I would like to thank you for your attention.
Did you find any dormancy, please? Any seed dormancy did you find?
I think it's apparent that seed dormancy was involved because compared to a fresh seed, dried seed, putting the seed into storage, the germination rate improved, so this is a clear indication that dormancy was involved. And storage for some period of time reduces the dormancy of the seed. Please join me in thanking Dr. Ebert. Thank you.